Hello everyone, my name is Zachary, and today I'm going to present to you my model organism, Schizosaccharomyces pombe, also known as fission yeast. So this is my overview of my presentation. We're going to talk about introductions, major topics of research, similarities and differences of these selected research topics, conclusions, and if we have time, some questions. So to introduce S. pombe, it's a unicellular organism with rod-shaped cells, and the rod measurements are very microscopic. It was first discovered in 1890, and was thoroughly researched after the 1950s. By this early research, scientists were able to conclude that S. pombe has around 1,200 different genes in its genome. And research on S. pombe has taken place all throughout the world, through Japan, Europe, and finally in North America. So some of the major focuses of my study is going to be on two major topics: genotoxicity, which is a term in genetics that is used to describe how chemical agents have effects on DNA and how they can damage, thus and creating mutations to occur. And then the second one is winemaking, where different cultures of yeast are used as fermentation agents to aid in the production of alcoholic beverages. And different strains of yeast are able to provide different aromas and tastes. So some of the examples are as follows: genotoxicity research in Bloom's and Werner's syndrome, water potability, which is what I will be conducting this study on, chromosome dynamics, fetal development diseases. And for winemaking, there is determining whether or not non-Saccharomyces yeasts produce higher quality wines, polysaccharides released during fermentation of alcohols, and alternative methods to traditional ways of producing red wines. So for the first one, it is genotoxicity studies with special reference to DNA damage by comment assay in fission yeast, Schizosaccharomyces pombe, exposed to drinking water. So this study was conducted in India, and as we all know, water is very important. Without it, we cannot survive. So the potability of water is super important to us. However, contaminants can get into our water, such as viruses, bacteria, things of that nature, and these things need to be treated. Treated water provides us with very clear, better tasting, better quality water. And with this without it, we can become sick. But even with the treated water, we can still become sick. That's where this study comes in, by showing examples of genotoxicity, where we use S. pombe to discuss this. So the materials and methods was that water was collected from various sources within India. Fission yeast strains PR109 was used for the study, grown at 30 degrees Celsius. Fresh and healthy yeasts were stored in chilled BPS, phosphate buffered saline, and used in mini gel in preparation for single cell suspensions. These slides were then exposed to raw water, which was the negative control, distilled water, which was the lab control, drinking water, experimental, and pure acetamide solution of 0.2%, which was the positive control. The slides were then submerged into a lacing solution, which is as shown here, and then the DNA was unwound and stained for any sorts of damage that was found to the DNA and analyzed under a fluorescent microscope. So some of these results, where it shows the percent damage to DNA, as you can see, the negative control shows that there's only 20%, and same with the laboratory control. But the experimental, for both 1 hour after and 2 hours after, has significant differences. And also that the TM, which is the tail moment, which is a very complicated calculation, shows that the higher that level is, the more damage ends up being. This is just a graph that verifies those results, showing that. So essentially, positive control tests positive with large amounts of DNA damage. Negative control is negative with little DNA damage. Experimental control tested positive, and as we saw in the graphs, the DNA damage was very significant. Therefore, water samples taken from tap water have very high genotoxicity potency. So some similarities and differences between the articles that I have selected is that the cells were grown into colonies using very similar temperatures and incubation times from the Lynn O'Connor article, and unfortunately that was all I could find. But some differences is that other strains of yeast for S. pombe were used for different studies in comparison to the one strain for the study, and that genetics were used to determine whether or not genes from S. pombe could be used to study different genes affecting fetal development in humans. So for winemaking, I used the combined use of selected Saccharomyces pombe and Lysacina fermentellarians yeast strains as an alternative to traditional malolactic fermentation in red wine production. So essentially what all of that means is that non-Saccharomyces yeast is being used to produce red wine. And originally, traditional winemakers thought that this would produce off flavors and high levels of acidic acid, which just can upset stomachs and all of that. But within the modern day, we have better research and conclude that it can be a lot more beneficiary to try these studies. And S. pombe is just simply used to deacify acid, which converts the L-maltic acid into ethanol. And it also releases high concentrations of polysaccharides, providing better aging. So the materials and methods is that 3 main strains of yeast were used, and fermentation was using a must from Tempranillo grapes, and this must was pasteurized. All the yeasts were fed the same nutrition at 60 grams per hectoliter, and yeasts were inoculated and combined with the grape must, and then the sugar concentrations were given to 249 about grams per liter, and then the fermentation finally began. Once the sugar concentrations reached about 3 grams per liter, then the wines were racked and stabilized to halt any further fermentation processes. The wines were then evaluated by 15 experienced wine testers, who were 5 female and 10 male, who tested for taste, aromas, and the overall appearance of the wine. These are just some of the results. The first graph shows the yeast population after the KT declined after 14 days when it was combined with other yeasts. And SC, which was the Saccharomyces cerevisiae, and SK, which was the Schizosaccharomyces pombe, decreased slowly over the time. The second graph essentially shows the amount of sugars and other things that they tested, and from this it concluded that the SC consumed sugar the fastest. This graph just shows everything from here more in a numerical form. So overall, the non-Saccharomyces wines tested very well against the conventional Saccharomyces cerevisiae, and it did improve glycerol and pyruvic acids, but it did not increase the overall acidity. However, they did have reduced ethanol levels, so those trying to get drunk off these wines aren't going to have much success. So some similarities and differences is that 3 of my articles ended up being from the 3 same authors, which showed their own progression within their research on this. And they all used the same methods, but came to different conclusions as you would by doing more of the same research. Another article agrees with the first one, that S. pombe has higher tendencies to release polysaccharides in comparison to S. cerevisiae, and wine samples were produced from the must of the same grapes, Vitis vinifera L, from the De Fiorenzo et al. So now some differences is that in the 2016 study from those that shared the same authors, it tested for higher ethanol values than in 2015, and then in 2017 they furthered their research by adding citric acid concentrations and urea concentrations to see if there was any significant differences. And finally, fermentation trials were conducted at different temperatures within one of the articles. So overall conclusions is that S. pombe is a very versatile yeast used in both medicinal and beverage research studies. And through the research of alcoholic drinks, humans are able to rest and recreation a lot better, having better tasting quality wines than perhaps more expensive S. cerevisiae wines. And within the fields of medicine, S. pombe's DNA can be used to determine the potability of water and research on harmful diseases to human beings rather than using animal studies or even human beings to test. So some special notes, if anybody wants to look at these articles, here they are, and then my 3 slides of references. Do we have any questions? Oh, yes, Don.
You mentioned that 3 of your articles were by the same author or authors. Did you see any similarities or slight differences in their research methodology, or did they use the same?
They essentially did use the same amounts of yeast. They used the same must for the grapes. The fermentations were all the same, but they tried to calculate for different for different numbers and such.
I guess what I'm asking, sorry if I worded badly, is did they use different samples for each one, or did they use one sample and publish 3 papers out of it?
They did use 3 different samples, but over the course of 3 years, and for the last one, they just decided to test for more values with the urea and the citric acid. And then the last one the second one, the 2016, ended up having more ethanol values. So since there's a difference between 2015 and 2016, do a third one to see if there's any more consistency or such. Anything else?
So you mentioned that medical research also with the Saccharomyces. Could you tell us a little bit more about it? Because I don't think you showed it, right?
Mm-hmm. So moving back a little bit to my examples, I discussed how fetal development diseases can take place. And so what Schistosaccharomyces pombe uses to do this is its own genetic information can be tested in comparison to human fetal cells, and they compare the DNAs and see if certain diseases can be arised, because the S. pombe can be used to determine whether these things are on the rise, and just compare those statistics towards humans.
Oh, okay, thank you.
You're welcome. Out or?
Let me do it.
Okay.
Guess it's for Philippine.
And it takes long time.